And so we get to our last video. Chapter 21 will make up the bulk of this video. This video is, or this chapter rather, is about the evolution of populations. And then we'll do just a little teeny bit of chapter 22. Chapter 22 is all about speciation, and if you continue on in Biology 102, you will start with chapter 22. So we'll do just a little bit of 22 as sort of an end cap on kind of the final step in evolution, which is the production of a new species. So in this chapter, we're going to talk about essentially microevolution, which is how a population changes over time. So we have two definitions of evolution. In chapter 19, we saw the idea of descent with modification. And this, of course, happens through natural selection. Living species are descendants of and so share characteristics with ancestral species, but they're also different because natural selection has made them different over time. So they are descended from but different from their related ancestral species, which is how we get this huge diversity of living organisms on Earth. Our present definition is a little bit different. It's not to say that Darwin's is wrong. It absolutely is not wrong. It's just a different way of looking at evolution. Our present definition is that evolution is a change in the genetic makeup of a population over time. So this is really just linking Charles Darwin's ideas with what we know now about genetics and inheritance. So we're going to look at how populations change based on the quantity and the frequency of alleles within a population. Natural selection is going to work on those alleles, and it's going to change the frequencies of those alleles over generations. And that is how we see evolution today. So evolution can occur in a couple of different ways. Evolution is a change in the genetic material in a population. So this can be because of mutation. This can be because of selection. And there are many things that actually affect evolutionary change in a population. We talked about natural selection quite extensively in chapter 19, but that's not the only way that evolution occurs. It's not the only way that traits can change, the frequency of traits change in a population. Chance events can also affect what happens in a population. So if by chance all of the birds of a particular color die in one generation due to, say, a drought, and that drought isn't choosing to pick a specific color of bird, it just happens that all of those of a particular color happen to die, along with others as well. You have now observed a change in that population, but not due to natural selection, due to some sort of chance event. So there are a couple of different types of chance events that can affect the evolution of a population, as well as any selection pressures that happen to be occurring in the environment. So there are two types of evolution, sort of two different scales of evolution. The first is microevolution. So micro means small. So if you think about what this means, microevolution is the evolution of a small group of organisms. It's the evolution or the change of a population, how the characteristics change in a population over time. And you can measure this by determining the frequency of all of the alleles that you are observing for a particular trait. So this might be the allele for red flower color, or pink flower color, or white flower color. And so you can observe how over time those allele frequencies change, how some years red flower color is more prevalent, some years pink is more prevalent, maybe the white completely dies out one year. All of those are changes over time. One important thing to remember that I should have said when we were talking about natural selection and chance events is that evolution is not always directed. So natural selection is sort of directive. It selects for those traits that are most advantageous. But evolution as a whole is not directive. You can have change without picking a specific trait. You can have change due to chance. And sometimes students forget this, that evolution can occur without natural selection. There are other ways, other mechanisms that can cause evolution besides natural selection. So we're going to look at how those other causes of evolution and natural selection as well affect the allele frequencies for a particular trait. So whatever trait you're looking at can change from generation to generation. So we're going to actually use a method to quantify this change, to be able to look at a population and say there were 3/4 of the population had this allele in this generation, and in the next generation only 1/4 had them. Why? What happened? What caused this change? So as an example of microevolution, the change in a population over time, we're going to go back to our ground finches. So these are the same finches that Darwin studied quite extensively in the Galapagos Islands. But in 1977, a group of researchers were studying these finches and looking at their diversity and how they change. And lucky for them, they experienced the Galapagos, I should say lucky for the researchers, not for the birds. For the researchers, the Galapagos Islands experienced a severe drought in 1977. And this was sort of a lucky coincidence for these researchers because this provided an opportunity for them to be able to observe a before and after population centered around a really important event that they could say, look, this change happened after this event. And so what happened was there was a severe drought. And when there's a severe drought, a lot of small, soft seeds will dry out. And if they dry out and are ruined, obviously you can't produce the next generation of seeds. And so the seeds that do survive are harder, larger seeds. And so if you remember, one of the things that Darwin observed about the finches was their beak shape, their beak size and shape. So these finches that are eating small, soft seeds are going to have small beaks. This guy here has kind of a small beak, not a super tiny one, but those beaks that are on birds that are eating larger seeds are going to be much larger. They're going to be stronger. They're going to be more robust. So this severe drought happened in 1977, and they noticed that after the drought, there was a lack of small, soft seeds. So if you think about for a minute what this might do to the population, well, if small, soft seeds are no longer available, any bird that eats small, soft seeds is going to have a hard time finding food, and so they're going to die out. So this is a really good example of natural selection working on a specific trait. If you have small, soft beaks, small beaks, there we go, small beaks, in an environment where there's no food, selection is going to act against you. You're not going to survive. You're not going to produce offspring. You're not going to pass those small beaks on to the next generation. But if there are no small soft seeds, but there are large hard seeds, and you have a beak already that can crack those seeds, you're going to fare better than those with the small beaks. And so you're going to pass on your genetic information that codes for those beaks. And in the next generation, you're going to see a change from small beak population to larger beak population. So those individuals that have larger beaks are going to represent a larger proportion of the population. So that's exactly what they saw. In 1976, the average beak depth was roughly 9 millimeters, and that was similar to several years prior. Then the drought happened in 1977. Only slightly more than 10% of the birds survived, 180 out of 1,200 birds. And they noticed that in the next generation of birds, they had a significantly larger beak size, almost 10 millimeters. So this was really important because it not only showed change over time, but it showed change associated with a very specific event, which was the severe drought. So that's our example of microevolution, how a population can change over the course of even just 1 or 2 generations. Our second type of evolution is macroevolution. So if you think about what that means, macro means large. So this is evolution on a large scale. And this is probably what you think of or what you're familiar with when you think of evolution. This is the production of new species. So usually the fossil record is studded with these major evolutionary events, and there's all of a sudden a new organism is found in the fossil record. That's usually what people think of when they think of evolution. In reality, the evolution that we see now on our time scale is microevolution, how populations are changing. But if the differences in a population become too great, that the individuals cannot interbreed, you actually can have the formation of a new species. And the formation of new species depends on how the populations interact. And we'll actually come back to this. This is chapter 22, and I'll talk about the two major types of speciation, which are called allopatric and sympatric. And those are just the ways that populations can or cannot interact, and therefore affect how new species come about. So we are going to use population genetics to study evolution. And so we are going to look at how the role that genetic variation plays in a population. So if you're thinking about macroevolution, you're not really looking at the specific allele frequencies. You're looking at the big changes, the changes that make one population so different from another that they cannot interbreed anymore and are no longer different species. I'm sorry, no longer the same population and are now different species. So our definition of, or our study of microevolution and changes in a population really depends on how we define a population. So a population is strictly defined as a group of individuals of the same species interbreeding in a specific area. So there are sort of three prerequisites for a population. So you can define the area as anything you want. The area could be the entire United States if you want. Generally, though, it's talking about a region, a smaller area. But another important characteristic is that they have to interbreed. So this is a group of organisms that is interbreeding. They are physically, meaning in the same area, physically able to breed. They can interact with each other in order to breed, and they're the same species. So however you define a population, you want to be very specific in what area, what region you're talking about. Now, two populations may interact or they may not interact. This map here shows two different populations of caribou. One is called the porcupine herd. You can see their range there in pink, and then the 40-mile herd range is shown in blue there. So this area is up on the Alaska and Canada border, way up very far north. And you can see that there is actually some overlap in range. And so these populations could potentially interact. They could potentially interbreed. I don't know if these specific caribou are interbreeding. And it's okay if they interbreed. They don't. Two populations can interbreed, and that's actually really important in evolution and changing population gene frequencies if they are interbreeding. But you can also see that some of the range does not overlap. So there may be individuals in the far north or far south that are not interbreeding with the other population. The next thing that we are interested in if we are studying a population is the gene pool. So the gene pool is determined by the population. The gene pool is simply all of the genetic information in that population. So this means all of the alleles for all of the genes that this population carries. And it's actually the gene pool that changes as evolution occurs. The most important prerequisite for evolution to occur is variation in the gene pool, variation in the population. If there is no variation for a particular allele, no one can survive better or worse than anyone else. And therefore, evolution cannot occur, because remember, we're talking about evolution now as a change in allele frequencies. So if everybody has the same allele, nothing can change. You might have less individuals with that allele, but the frequency of the allele is still the same. So variation is absolutely required at a particular allele if anyone is going to, if that population is going to evolve, if anyone is going to survive better than anybody else. So as we said in chapter 19, people have been using artificial selection for a long time. They have been selecting for variation that they think is important in whatever they are interested in. But variation, if we're talking about evolution, has to have a genetic basis. And so even in a natural population where evolution is working on a trait instead of people, that variation, again, must have a genetic basis. So there has to be variation, and there has to be variation in the genome. So there has to be a genetic basis for whatever allele you're going to look at as far as evolution is concerned. I think this picture on the bottom is funny. It's a Great Dane with a little tiny dog of some sort. But this is just to show how wide the variation can be in a species. So these two dogs are technically the same species, but if you weren't familiar with dogs, you might call them something different because they are quite different. All right, so let's talk about where this genetic variation comes from. Now, none of this is going to be new. We've talked about all of this before. So this chapter is really sort of a good review of and synthesis chapter of things that we have talked about. Sort of brings together why all of that, the genetics we went through, the molecular biology, why all of that is important, not only for function of a particular organism or reproduction, but why it's important at the population level. So sexual reproduction is really important in keeping genetic variation in a population. Sexual reproduction is a really great way to shuffle alleles and make new combinations. So we talked about crossing over, where two sets of sister chromatids will exchange genetic information. So chromosomes from mom and dad will swap alleles and make new combos. Independent assortment, so that is arrangement and combinations of whole chromosomes. And then, of course, we have random fertilization, where you have hopefully two unrelated individuals bringing their unique genetic combinations together to make something completely new. So sexual reproduction is probably the most important source of genetic variation in a population. Organisms who reproduce asexually actually do have some means of increasing their genetic variation. So bacteria are a good example. They don't reproduce sexually, but they can actually exchange some genetic information. And again, that's something that you go into if you take 102. So asexual reproduction is very, very important for sort of maintaining this high level of variation and making new variation every generation. So mutations are also really important in genetic variation in a population. And mutations are important because it's the only way to produce new genetic variation. And I'm pretty sure that I have said this before, but the reason that mutations are important is that they can produce new alleles and therefore new phenotypes. So these are going to be different phenotypes that weren't present before. When a mutation occurs, it can have one of three effects. It can be neutral, which means that the mutation has no effect whatsoever on the survival of the individual. Now, this might be because it is a silent mutation where it doesn't change the amino acid, or it might just be a mutation that actually causes a phenotype difference but doesn't actually affect the survival of an individual at all. Maybe it's a slightly lighter color of tan for a mouse, or maybe it's slightly longer fingers. I don't know. It can be anything that individual does not give that individual any sort of selective advantage. Of course, the new allele can be advantageous, which means that it does give them some sort of edge for survival and reproduction over others in the population. And then at that point, you will see positive selection. The allele will, for that new allele, will increase in frequency over time because individuals with that allele are reproducing and passing it on. Most alleles, though, if they're not neutral, are going to be detrimental. And this is because, although natural selection and evolution are not perfect, they are constantly working to make sure that an organism is going to survive. So if you have an organism that comes up with a mutation or even a new combo from sexual reproduction that is disadvantageous, a new allele or a new combo of alleles that makes an individual different is probably going to be detrimental because that population should already be perfectly adapted to the situation they're in. And so if they have a new allele that comes up, it's probably actually going to be a problem for that individual. They're probably going to have a lower survival rate or lower reproductive rate than others because of that new detrimental allele. But keep in mind, they don't have to be. Sometimes they are advantageous, and when they're advantageous, it's great for the population. Mutations are important when they're in the germline. So what that means is they're important when they're in the sex cells, the sperm and the egg. Mutations that are in the somatic cells are not passed on to the next generation. So although they're really important, they're not important for population genetics. So we are worried about those mutations that get into the sperm or the eggs and are then passed on to the next generation. Another important thing is that recessive mutations can be hidden. So remember, we have heterozygote individuals. And so sometimes you might have a mutation come up that you don't actually see in the population because when it's in really low frequencies and they're in the heterozygote, you might not see that mutation for many generations until an individual happens to have the homozygous recessive. So sometimes allele frequencies don't even actually affect a population. Sometimes mutations don't even affect a population's allele frequencies for quite some time. Our last source of genetic variation is altering gene number or position. So we talked about this, I think it was chapter 12. This was changes to the chromosome. So duplication or deletion of a region of chromosome, inversion where we flip-flop a bit of the chromosome and stick it on backwards, translocation where we move a section of a chromosome to another one. So these are important because they can change the way that genes are regulated. They can change their function. Obviously, if something is deleted, a gene is now completely missing. But it's not always a bad thing. Sometimes you have problems in crossing over in meiosis, and the resulting duplication is actually a good thing. The reason is that if you duplicate a region and the old part remains intact, that old part can continue to do its job, but the duplicated part can actually take on new functions. So an example of this is gene duplication in mice and humans. In the olfactory genes, or the genes for smelling. So humans have decent sense of smell, not fantastic. We have a small number of olfactory genes, but mice have something crazy like 1,200 genes, olfactory genes, and that is due to gene duplication. So they kept their original genes, and then when duplication occurred, some of those genes could enhance the old one. So now you have two genes doing the same job. Or multiple genes doing the same job. And so that was really advantageous for this mouse, because if you are constantly trying to stay away from predators and you have a really good sense of smell, you're going to have an advantage over your buddy mouse who may not have that duplication. So something important to keep in mind, and I said this before in chapter 19, is that individuals do not evolve. Individuals do not choose their adaptations. They are born with whatever they have. So a population is determined by whatever the individuals are born with. But the population can change with respect to the number of individuals that have that trait. Natural selection works by altering reproductive success. And so what that means is that you might observe that an individual dies, or you might observe that they only pass on, or they only have one offspring, whereas someone else has four. But you can't see the effects until you look at the population level, until you look at what that individual dying or leaving only one offspring is actually doing to the population as a whole.